Holmes from Stanford University. Susan has had several contributions to the area of phylogenetic tree, community interaction graphs, and metabolic networks. I personally remember when I was doing my PhD, I had to code this Markov chain Monte Carlo sampler to estimate a phylogenetic model. And that meant that I had to uniquely label phylogenetic trees, and the common thing to do was to use this really complicated parentheses, nested parentheses notation that tries to to identify unique, or what identifies uniquely trees. But back then, my computational skills, programming skills, were not that good. So it rescued me really to find one of Suzanne's papers, a really elegantly written brief paper on how to uniquely identify a binary tree by an integer number. So my problem turned from having to code something made up of parentheses, semicolons, colons, and characters, to having to code an integer number, really, to identify, to count trees along my Markov chain. So Suzanne specializes in mining messy biological data sets, and she's currently working on image analysis, drug resistance in HIV and microbiome data analysis. And I think her talk is really relevant to this meeting. I have personally been asked in the past what is the best thing to do in terms of normalizing metagenomic data sets. So I'm really looking forward to hearing what Suzanne has to tell us. Welcome, Suzanne.
Thank you very much. Well, I thank the organizers for inviting me, especially because I'm sending my sabbatical over in Toulouse, just over the water, and it's much closer and easier than coming from Stanford, so I'm very happy to be able to spend time here. And in particular, I'll be here for 4 weeks, so if there are things that you'd like to ask me more about, I'm happy to elaborate. The document is a PDF document, and there's a lot of links which are live. Mostly things which are read are alive, so you might be able to find more details there. I'll try and show you how that works. So I like to say I've been working in biology since about 1985, where I specialized in the bootstrap, which meant you could squeeze out extra data from a data set that was too small. And then we started to have microarrays in 1998, and that's the fire hydrant, so we really had a lot of data. And then around 2012, we have this sort of perfect storm. We have data coming in from every side. Now, I'm not going to stand up and say, you know, my data is bigger than your data. That's what the men do. I'm going to say my data is much more heterogeneous and complicated than your data. That's what the women do. And so in my case, the data are heterogeneous. They are heteroscedastic, which is a sort of micro version of heterogeneous, which we'll go into. We have structured high dimensionality, and I'll show you some examples of what I mean by that. And we have to integrate both graphs and trees. We have information leaks, which is a problem, and we have reproducibility and validation, which is also a problem. So I'll start with heterogeneity, and heterogeneous data are all alike. Homogeneous data are all alike, and all heterogeneous data are heterogeneous in their own way. So what do I mean when I talk about heterogeneity of the type of data that we're looking at? First of all, sometimes we know what we're trying to predict, some kind of illness or not. And we work on, I've worked on IBD, I've worked on various other kinds of control. You have control healthy individuals and you have sick individuals, and then you want to try and, I call that supervised, non-supervised, and sometimes you have a response and explanatory variables. Sometimes the variables aren't measured. So you have, and we've seen a lot of that, people trying to find clusters. What is happening is there's a hidden categorical variable which you haven't measured, but you need to find it. sometimes there's a hidden gradient, which I've worked a lot on, and that's a hidden continuous variable. Sometimes you have just mixture of types, and so we have graph information and tree information, which is very important, but we also have, we've worked on the rat IBD model, and we had samples along the gut, so we have spatial information. Sometimes we have spatial information because we have images, and then you come up against the problem that you have different amounts of dependency, so you also have that level of heterogeneity. And of course, all of you working in the field know very well that the different technologies involved also give you a level of heterogeneity. So what are we going to look at? And I'm not going to give very many details. There's the microbiome on the one hand, the metagenome on the other, which is all the genes, and then the transcriptome, or the metatranscriptome, which I'm also very interested in. And so, of course, you will have seen many pictures like that. That's how I got interested in this. I started working in the field around 2004 because I like phylogenetic trees, but I wanted to do some applied work. And so the data we're working with, and mostly I work with David Relman at Stanford on this, and have been for quite a while, are of very many different types. And so when you have very many heterogeneous tables, you have a problem of comparing them and keeping them together at the same time as normalizing within the tables. So what are the data? Well, we have DNA data, sometimes just the 16 sRNA. Some of it, which we've looked at, also has the metagenome, the type of data we've been talking a lot about. Sometimes we have expression through RNA-seq, which we're trying to use to see what's being expressed and do a functional analysis. And we also have mass spec data, so we know something about the chemicals that are there, although the data is very sparse in annotation. So maybe we can annotate only about a third of what's there. We have clinical data, which is extremely important about everything about the patients, and sometimes environmental data, and a lot of domain knowledge. And so the secret in doing well in data analysis is to keep all the data together as much as possible at the same time. And in order to do this, we've created a system within R. It's a bioconductor package called PhyloSec, and it enables you to manipulate very easily heterogeneous data objects. So if you wanted to take a subset, for instance, of only certain samples, it'll take a subset and that will repercute all the way through the different pieces and simplify your tree, for instance, if you drop some of the taxa. So it makes it very easy to manipulate. So we use this object-oriented framework in R to do that, and we have matrices, data frames, and trees that we code. And for that we use APE, which uses that matching representation, which is parsimonious. And we have to interact with a lot of players. So we work a lot with, of course, people in biomedical science, but also with ecologists and people who have I work with Les Defselson, and although we work on antibiotics, he has a training as an ecologist, and it's very useful to come in with that training compared to, but these are not standard clinical trials, so biostatistics doesn't prepare you so well for this type of data. But as you see, genetics and bioinformatics are very important, but visualization is important too, and mathematics. So here, of course, the standard pipeline that we've seen a lot of, and I'd say that whatever you use, for instance, to cluster your OTUs, then you need to import the data into R. So we've specialized in the bridge part, and so we have many importers which allow you to do that. And the representation I like to think about mathematically, even if it's not the most sparse for graphs and for trees, is still to use matrices. So we think about things as matrices, and very often the tree, the network, will be linked to a certain kind of weighting or a certain kind of distance metric. So this is what a pipeline, say the word, you start with rectangular data and a lot of side information, and then you import it. And when you import it, then you have the possibility after getting the raw data, you can do pre-processing. And pre-processing is very similar. If any of you analyzed microarrays, you had to filter out genes, for instance, and filter the data and prune the data, and all the pre-processing is done here. And then you have to make a lot of choices. There are 75 different distances you could choose before you did the ordination. And so choices come up, and they're very important. You can plot the heat map, you can plot networks, and then you can do inference. So there are all kinds of packages which already exist in R. For instance, for choosing the number of clusters, you can use a package which has the gap statistic for specifically choosing the number of clusters automatically. You can do multiple testing correction and things like that. So that's the advantage of having a system like that. And this is what the data looks like. So this is an example of the data set which is in the package already. It's the global patents data from Caporoso. And you can see this multiple object is here. And here is an important thing. This is what we call a table, and it's just a matrix with counts. And then you have the sum of the counts for each of, these are all samples, and in rows you have, for instance, the OTUs or the genes. And these counts here will be very important to us, and we'll use them to do some of our normalization. But one thing you can see is they vary by orders of magnitude. So the sum on this column is 59,000, and sometimes it goes all the way down to 2,000, the number of reads. So for a statistician, when analyzing this data is called a contingency table, and that's what we're going to analyze. So if you want to look a little bit more, that's the wiki, for instance, is there. And you can look on, all the different examples and how to import data, and that's alive. So that will be useful. Let me go back to my where I was, and I want to talk about the heterogeneous problem now. So how to normalize the data. So we learned early on in doing microarrays that the kind of data that we collect is not normally distributed. Here, this is a Laplace distribution, which is due to being a large mixture of many normals. It's what we call a scale mixture of normals. And the mixture that we have to deal with is one in which you have, for instance, the library sizes which are different. So the columns of the contingency table are all different values. So how do you deal with a different number of reads? Some of the ecologists were inspired by work that was done by Saunders in 1968, which was a very clever paper because it predates the bootstrap. So it means that Saunders had the idea of the bootstrap before the bootstrap was invented by Efron. And the idea here was to take subsamples of the data in order to make rarefaction curves. Unfortunately, the ecologists liked this method too much, and now, in order to normalize the columns, they have a procedure which is based on rarefying. So taking columns and taking a subsample of the data. So the procedure, for instance, in Chime, but it's in many of and Mother as well, is you select a minimum column sum, and for all the columns, all the samples which have smaller than that, you drop them. And for the others, you take a subsample of that size, and then you have all the columns will be the same size. So that's a problem. If you do it differently, if you reduce your data to proportions, you're also losing information. And so as a statistician, every number you see, you have to think of it as coming with a little interval around it. But if you just reduce all your data to proportions, you just lost that. Now we have a way of saying losing that information isn't acceptable. We say it leads to inadmissible procedures. So procedures are inadmissible if you can always beat them. So dropping data is inadmissible if the data isn't useful. So we have a way of saying whether the data is useful or not. And if you have the original data and you, for instance, you have a pipeline and you're squeezing out, you're taking less data, you're going to have a leak of information, and your final results might be very, very not strong enough, very light compared to what you could have done with your original data. So for instance, when you're looking at a binomial distribution, a proportion, the proportion is not a sufficient statistic. It doesn't contain all the information in the data. If you know the number of trials, then it is a sufficient statistic. So the sufficient statistic is the couple NP, not just the proportion. And what do I mean by sufficiency? Well, you can translate it into standard statistics by saying that the joint probability distribution of the data, conditional on the value of the sufficient statistic, is going to be the same as the distribution conditional on the original data. And it doesn't depend on the parameter, but you can also use an information measurement and look at mutual information. And in some sense, the mutual information about theta, for instance, if theta is what you're interested in, and you suppose that you have a distribution on the thetas, then the mutual information here is the same. That is, all the information in the sample, or in your random variable, is contained in the sufficient statistic. So you need to use a sufficient statistic. And for a Bayesian, of course, they have a very nice definition, because they say that no matter what prior you use, the posterior will be unchanged whether you use the sufficient statistic or the full data. So you only need the data through the sufficient statistic to compute the posterior. So always choosing a sufficient statistic is a principled way of deciding how to compress your data. So many of these studies that we're doing now are in the clinical setting. And in the clinical setting, you want to decide whether the healthy subjects have more of certain genera or species than the controls. And we have certain standards for doing this kind of clinical study. And one of them is it has to be optimal. That is, you have to use the data as well as possible. And so you have to have the highest power possible given your resources. So it's a little bit what I was talking about yesterday when we talked about how do you design your experiments. Well, you have to start off by saying how many dollars you have to spend, and then you have to decide how you spend them. And so how do you measure how well you're spending them? Well, you want to maximize your sensitivity, or the power, which is the true positive rate. And sometimes you want to also, but they counter each other a little bit, the specificity. So we have to control for many sources of error, and we have to maximize that. So, as I say, I want to say why you have a decrease in power if you change your original data to take a subsample of the data, and this is just a little example. We're going to come to a more sophisticated way of approaching this, but this is just to make the point that if your original abundance is here, you have this column has 100 and this one has 1,000, and you do rarefaction, and here you subsampled and you got the most likely subsample, which is 50-50, and you do a test. In one case, you have a p-value on the original one, whatever test you use, which is smaller than 5%, so the clinician you're working with will be happy. P-values have to be smaller than 5%. I don't like p-values. But if you use the rarefied data, you can't conclude. So you didn't use the data well enough, and so you lost power. So losing power, even if you have a lot of data, that's a problem. So what do we do? Well, we want to try, the idea behind rarefaction was you want to try and make it so that everybody has the same variance. So you want to try and equalize the variance, and that's done by what's called variance stabilization. So we take transformations of the data. Now, some of you have already, in your talks, done things like this because you saw some of the time square roots, some of the time there was a log taken. So there's a feeling that you have to transform the data. This is just a principled way of doing it. And so to quote Daniel, mixture modeling works miracles. We use mixture models. They work very well for this kind of thing. And I put up a link here to why mixtures are really important in statistics, the Finetti's theorem. The mixture that we're going to use is the gamma-Poisson mixture. So in that, we'll have a parameter that we generate from the gamma distribution. And it's like a hierarchical model. You start by generating the parameter, and then you generate from a Poisson with that parameter. And it turns out that this negative binomial, or gamma-Poisson, fits the data very well. You can do better with a more complex model, but we found it sufficient to first order for doing a lot of things. So this corresponds to the variance stabilization. You fit this model to the data. And so here's a picture from Wolfgang Huber, which explains a little bit what you want to do. If your perfect variance stabilization in the case of the Poisson is to take a square root. If you take a square root of a Poisson variable, you get a constant variance. In our case, the data aren't Poisson unless you're only looking at technical replicates. If you have added noise, you have to have a more complicated model, which is this gamma-Poisson mixture. But still, you can aim to have a transformation here, the idea being that this is the original data, x here, here. And you see that we have a lot more variability at the high end. And we want to make a transformation so that every number comes with an interval, which is of the same size. That means that everybody will have the same weight. And it'll be important whether or not you're doing principal coordinate analysis or whether you're doing testing. If every point has the same weight, you can look at the plots quite simply. If some of the points are very, very fuzzy, when it comes to variance, those points will, if you use a PCA method, you're trying to maximize the variance explained, those points will pull your study. So you need to have everybody on the same scale. So we model, and we use this Poisson gamma model, in which, in fact, we have meaningful parameters. So if you had technical replicates and they had the same number of reads, and I call that Sj, that's going to correspond in some sense to the column J, and then you have a incidence of a certain taxoid, so your rho, you have a proportion which is ui. Then your final measurement, in some sense, will be Poisson around this. So that's what you would have if you just had technical replicates and you had exactly the same number of reads. You'd have some noise, and we say it's a Poisson noise, and then you have this. Now, to this model, we're going to add extra effects. Because of course you have the different replicates are not technical replicates, they're biological ones. And so we're going to model the counts with a more complicated model. And in this more complicated model, we add extra variation with a parameter here, which corresponds both to the sample condition and the OTU, for instance. So we have this model, and this model allows us, in some sense, to have enough freedom, and we can estimate it by borrowing strength across the whole table, and we can normalize the data using it. So this allows for doing, this is a hierarchical mixture model, and it's very similar to what happens in standard analysis of variance, where you have a random effects model. So we know how to do testing and things like that. So we used an implementation, we carried over, this is a paper which is going to appear next week in PLOS Computational Biology. It's called Waste Not, Want Not, Why Rarefying Microbiome Data is Inadmissible. And we have in the paper all the different steps are documented in R, so you can go through every step that we did and every picture and every simulation that we did and do it yourself, and then you can do it on your own data. In order to look at the over-dispersion, the fact that we have the variance is not the variance of a Poisson. In a Poisson, you have the mean and the variance are equal. We looked at several data sets. In the supplementary material, we have 6 data sets. I'm just going to show you 2 of them, just to prove that this fits pretty well. So these curves here, this curve is what you'd get if you did the Poisson. And this is the curve that you get if you do the fitting of the negative binomial. So we get a curve where we have the variance as a function of the mean, and you can see that none of these are Poisson. And we have a pretty good fit, although we could have enriched it a little bit by making something zero inflated. We didn't, we just tried to keep it simple. Now, of course, when you had the rarefied data, you have much less data points, but you still have the same phenomena that it's not Poisson. So we see an improvement in power and in the false discovery rate. And we did various simulations and comparisons on studies where we knew the answer. So these are just to show you an idea of what you get. So we use the area under the ROC curve, which is called AUC, area under the curve, and we have the specificity and the sensitivity of the different tests. So we compared different approaches to this. There's a normalization process using a normal, which is available in metagenomics. There's a 2-sided Welsh t-test. You can't always do it. That is, if you're just trying to compare proportions and you don't know the number of trials, you can't do that. So some of the data didn't allow us to do that. But we were able to see that you have although the model isn't the right model for the data, it still gives you better results using this negative binomial. So both DSEC and EdgeR are implementations which exist in R already for doing the negative binomial tests. So we did a transformation of the data and put it in that. So these programs were developed by people who work in RNA-seq. So in RNA-seq, you very often have this problem of the library sizes being different and various sources of error. I'd say you have an additive source of error and a multiplicative source of error, and you have to combine them somehow. So those are well programmed. The other thing that it does is it also improves when you're not trying to do differential abundance testing, but you're just trying to cluster. So we see also improvements when you're clustering. Here we did a simulation experiment where we took templates from 2 different sources on the global patterns data. We took, we took the fecal and the ocean microbiomes in the global patterns data, and we used those as our templates for mixtures, and we made the effect size bigger and bigger and bigger to see who could detect the clusters the best. And we used different distances and different normalizations. And in this case, we did, clustering using medoids, the PAM implementation, but it's partitioning using medoids. And we look at the accuracy, and what you can see is, in fact, in this case, proportions, for instance, do quite well. You don't have any evaluation of the quality, but if you just use proportions on the data, they do quite well. And so you just have the plain proportions. It's certainly the case that if you rarefy here, your accuracy goes way down. And so here the proportion plateaus here, and it has to do with several things, but one of the things is when you do rarefying, you discard some of your data. So discarding some of your samples loses you power, loses you the ability. So we have the simulation details. I want to go on a little bit, so I'm not going to. Okay, so now the next question, once we've done this, is a question about keeping all the data together. So we have to keep as much data as relevant, so we have to use sufficiency. And we also have to keep the data together, and how do we use the metrics? So principal components, as it stands, is not the right method. The ecologists understood this very early on, and they have an adaptation of principal components called correspondence analysis, where they generalize. It's a generalized singular value decomposition, but here the metrics, they have special metrics D and Q, which take into account the row sums and the column sums. So correspondence analysis doesn't work on proportions. And so you need the full data, you need the counts, you need a contingency table. But if you have that, it works pretty well. And you can do constrained correspondence analysis and various versions the ecologists have developed. And so we made an interface to two ecological packages, vegan and AD4, which works pretty well. So for instance, this is an example where they do a correspondence analysis, and one of the most useful things in correspondence analysis is you can find gradients through finding arch patterns and horseshoes. So that's something that I use a lot, but in order to do it, you need a contingency table. PCOA, of course, is something that people like a lot in the field, and you can look at various versions of that, so principal coordinate analysis. And the thing is, you want to keep all the information, and you have many, many different choices. So you could do a principal coordinate analysis where you show the taxa relationship as well as showing the sample relationship. So the standard one is to show the samples using unifrac, but that's a loss of information. You can do everything. And you can subset, for instance, and you can make. So R allows you to do that in a very simple way. And you can even, here's an example on the global patterns data. And the way it was done was it's just 2 lines of code. And so you just plot it. And of course, we use the ggplot2 package for plotting because it allows you to layer the graphics. So you can keep adding information. You have one plot and then you just keep going. And we can put both the samples and the taxa, which is an added plus this biplot representation, which was first done by John Gower, is very useful for these kinds of studies. Of course, it was well known to the ecologists. They use it a lot. And so we try and leave this. Go back to not the appendix. There we are. So that's the... So how do we combine and compare trees? So I won't have time to do very much about this. There's a double principal coordinate analysis which allows you to take into account both the distance between the taxa, the abundances in the taxa, to create plots where you see distances between samples. And so this is something that we did quite early on, and we've compared it on antibiotic time course data. So when I first started on this project, the biologist had 5 sample of size 5. I hate working on samples of size 5. You can't prove anything. And then it turns out that we have metadata, metabolomic data, only on sample of size 3. So it's not really statistics anymore. Okay, so we're just doing visualization and talking about the data. We can't say very much about it. But now we were lucky enough to get a large transformative research grant from the NIH, and we're going to go back and we're doing this study, but instead of having 3 or 5 patients, we will have hundreds of patients. And on these patients, we're doing different types of perturbations. And one of them is we're giving them rounds of antibiotics, and we're looking at what happens, both on the microbiome, the metabolome, and metatranscriptome. So we're looking at many different levels on the same patients, and I think that's very important. So what you see is you can look at unifrac, for instance, or weighted unifrac, which is much better. You capture much more of the variability, or you could do absence presence. But what we did was we did a double principal coordinate plot, and the advantage was it gives you both the samples and it gives you all the taxa. So you can see the differences between the taxa and the differences between the samples at the same time. And it partitions the variance in a special way which takes into account the difference between the taxa on the tree. So that's here's some of the output. We put the patients before and after taking their round of antibiotics, and we called them stressed and non-stressed. And one of the things you can see is that there's a difference. So here, this is the data not stressed, and here this is after they took antibiotics, and here and here. And our hypothesis, what we're interested in, the vector here has a special meaning in terms of combinations of different bacteria, and we're trying to find out whether, with 3, it feels as if they're all kind of going in the same direction, but with 100 we'll be able to test that. So we'll be able to see whether that's true. So that's for the next. So the other thing that I wanted to talk about, and Hongxiang just talked about it a little bit, but I have a different take, but I agree with him completely. You don't do correlation networks. So correlation networks, some people were doing for various kinds of mutation studies. They were doing them for microarrays. They're full of artifacts. But something that we did was we did co-occurrence studies measuring the co-occurrence of bacteria using the Jacquard index, which is something that the ecologists like because it measures whether you have rare things co-occur count, whereas rare things not appearing doesn't count, whereas in a correlation that counts. So the Jacquard index is a better way of computing, making a network. So the first network that we wanted to do was actually not between which of the bacteria co-occurred, but it was which of the bacteria co-occur in different samples. So we have mice that we worked on, and these different mice are joined here with an edge if they shared more than 70% of the taxa, for instance. That's a typical sort of, and this is quite robust. Now, here, most of these are the same mouse. So we have a time course and we're looking over time. These are the same numbers, they're the same mouse. Now, if I go on to that there, and so we did have some cases where we had 71 with 72, 75 with 71. Those numbers meant something. It meant cage number 7 and mouse number 1 or mouse number 2. And what you see is which of course the biologist already knew, but is that the most important thing is not the diet, it's not the genetic makeup of the mouse, it's the fact that they live in the same cage. So that's also something that we saw because we didn't have enough power when we did a study of vegetarians and omnivores. And we had small sample sizes, 15 and 15, and our subjects were people from the lab and people across campus. They were volunteers, so they were people we captured. And it turned out that it was more important who you worked with than what you ate. Now, because there's a block effect is much stronger, in fact, than the diet effect. So it's true that you have to account for that. And here you could test it, and so we did just a permutation test in order to try and see whether you could retrieve a significance value using Friedman-Rafsky's minimum spanning tree test. And we got a p-value of 0.01. And so we also did co-occurrence, and here I labeled them only by the genera and not by the taxa. This is an unpublished work with a postdoc, and so I didn't want to say too much about the actual biology of the network. I wanted to take 5 minutes to say something about reproducibility and validation in this context. There's a complete sea change right now which is occurring in standard biostatistics, which is p-values don't mean anything when you have very large data sets. And we've seen it, we saw it there was a study about the enterotype of the human gut. People have had to retract a lot of different results because most of the findings, for instance, from microarrays haven't been re-verified. And so people are saying you can't use a p-value because you have too many degrees of freedom of how you're tuning what you do. So if you can choose the distance between, you have 70 choices of distance, you have 50 choices of clustering method, and we can, in the enterotype study, they dropped 13 samples, we don't know why. You can make anything appear. And so the problem is, it used to be p-value was the way you published, and it's not p-value anymore. It's reproducibility. That is that you have to put out there everything that you did to the data. What did you smooth? What choice did you make? And so you have to give a script. You give in the same way as when you're in the lab, you have a lab notebook and you keep a record of everything you did on the data, because that's integrity in science. It's the same for data. That is, any study I can give you a significant p-value. I'm a very good statistician and I know how to do it. Because I have too many degrees of freedom. It's too easy. We have too much data. So you can't use p-values on their own. You could use them a little bit, but you have to have measurements of error and you have to be upfront about all the choices that you make. So you have to show your code. So this is the enterotype, and for instance, if you look, this is just a little example on the enterotype data. So just using the enterotype data, we put it as an example data set in the Philosec package, and you just load it. And if you look at the different distances, so here we've colored by technology. We haven't colored, in this case, not by, we didn't color this by the enterotype. The shapes are the enterotypes in the next one. This is just to show you some of the distances. There are 70 choices of distances. You can choose the one you want, depending on what you're trying to prove. So you just, and this is 5 minutes of coding, right? So it's really easy. There's no effort involved. You just, this is the code. There's the code.
What are those different colors?
Sorry, what
are the different colors?
And so in this case, it's the sequence technology. So in this case, I colored according to sequence technology. They had 3 kinds of technology. And so, right. So what I have well, I'm on the record, so I don't know. So I have a hypothesis about what happened in that study. And but there are 3 groups, but they're technology. It's not so interesting. But it's just to say that there's a real difficulty in having clinical publications now which involve this type of data, and it's the same that happened for microarrays and RNA-seq, and people are changing the standards. So the standards for publication are much more about reproducibility, and you have to have the full code of what you do, and you have to provide it to the reader so that they can they have an idea of what you actually made as choices. And you can see you can often see whether there was a sweet spot, whether when you try to reproduce, where there was a specific combination of choice of distance and which just gave a result, but if you move something, it's not robust. So you can see that. That's sort of important. So you have these multiple testing. You're doing every combination, and you have to adjust. When you're doing multiple testing, we have ways of adjusting for the multiple taxa, but it's actually much more complicated. So I'm just going to finish with what we've done and where we are right now. We've done some modeling mixtures and we've used them for variance stabilizing transforms. And we have ways of combining heterogeneous data. And I haven't talked a lot about the multivariate methods involved, but you can ask me more. We have ways of computing with graphs and trees within the phylosec. And I'm thankful for people who've developed visualization programs like iGraph, which allow us to do that. We have also, you can do sensitivity tests through simulations in a couple of lines. And the supplementary material for the Waste Not, Want Not paper is available on the web already, and it contains all the different steps, and you can see how we did our simulations. You can reproduce them. So if you do anything by simulation, if you're trying to prove something by simulation, you have to set a seed for your random number generation so that the person can exactly reproduce what you did. And I think that's important more as we do more and more things with Monte Carlo. And multiple table methods, we have ways of generalizing the multivariate methods once you've normalized all the tables, so you can do multivariate time series. So I benefited a lot from all the tools from the R community that were already developed. And in particular, there were a group of ecologists in Lyon who developed ADE4. Wolfgang Huber and Michael Love did a great job on the RNA-seq normalization models, which were follow-up from what they did on microarrays before. And Emmanuel Paradis has developed APE so that you can do phylogenetics with R very easily, and I'm very thankful. I use RStudio, and I don't understand their business model because it's free, and it's a great IDE for computing. My main collaborators on the biology side are David, Alfred, and Les, and also Justin Sonnenberg. I've done some work on sparse methods from metabolomics, and Percy Diaconis, with whom I did a lot of the work on trees, the sort of theoretical representation. My postdoctoral fellows, though, Joey McMurdy has developed with me over the last 5 years. We've been working on ancestors of phylosec for a long time, so we have a final product which we published last year at +1, which is quite popular now. And Alex Alexienko, who's a postdoc in my lab, and Ben Callan and Angela, who does some of the metabolomics. And Elizabeth Purdom was the first student in the lab, and she did the science paper in 2005 with David Relman, and that's how all of this started, and funding. So that's all.
Thank you very much. Is there any question?
Yeah.
I thought you made a very interesting point at the end that, there's all these things you can do, and you've got every opportunity to fiddle the answer, and every temptation put in front of you.
Right.
So, and you mentioned also that you can tell when somebody's done this.
If you have the code. If you have the code and you see how they chose the tuning parameters, and you can rerun it and change the tuning parameter. And if it's just, they got this combination of tuning parameters, and every time you make a small change, you get a different result, then you know, right?
Yeah, so of course this will depend on the kind of analysis that you've got. I mean, if the investigators have used your package, then you can simply sort of load it all back in and just fiddle around.
Yeah.
You can even write a little script to do it all for you, and that would be fine. But on the other hand, if all you have is, as you say, they should provide with their paper the scripts that they've used or the decisions that they made, it's not necessarily so easy to explore the space of all these things.
No, I understand, but there's a problem. There's going to be a problem as soon as the FDA and the downstream, when you're going to start making clinical decisions, then they're going to want to know exactly what's your uncertainty on this. How robust is it? And so that's the issue. Yeah.
Taking this in a slightly different direction. I mean, I completely agree we have to sort of keep the scripts around, but doesn't it actually extend to the raw data as well? And all the transformations you're doing to
Absolutely.
from day number one? So, I mean, this is only one very small part of a major chain of things.
Yep.
And you know, if you don't keep the things in the beginning, stuff at the rest, in the end is just, you know.
No, I agree, but still, I mean, I'm talking about the statistics. So, because that's what I know very well, and I just know that it's very easy to prove something with the amount of data we have. We have too many degrees of freedom. And anybody knows, you know, p-values, when Jim Berger wrote a wonderful paper, but if you have a data set of 100,000 points, you always get a significant p-value. Even if the data came from something where you know that it was 50-50, you still get a significant p-value. So, you know, that's the problem.
I mean, it would be very nice if, without having to sort of run all the alternatives that you might have done, if there was some kind of thing, some kind of technique for sort of perturbing the whole thing in a very small way and being able to tell, oh yes, they've just sort of chosen something at the top of a curve, you know, without actually having...
A good paper has a sensitivity analysis in it and is honest about the uncertainty of the numbers that are input and says, okay, if I have this level of error, this is going to propagate this way, and my final result, you know, my confidence intervals like that. So, but that's, you know, best practices. You don't see a lot of... There's no incentive, by the way. There's no incentive because, for instance, for the enterotype paper, they didn't have to retract it. They only had to send in an addenda. They still have a Nature paper. So, you know, why? Why not? So.
But were the enterotypes the sequencing technology or not?
Originally, I think that's what they saw, and then they realized and they thought, oh, well, maybe we can. And so what they did is afterwards they went back and only looked at this in the subgroups within technology. But then they fiddled with the data. I'm going to leave out these 13 points because they're in between and they don't classify well. That doesn't sound right. So, but I mean, it's just one, it's not this particular problem, it's just an example of what happens if you have too much latitude in your choice. So they use Jensen and Shannon distance, and you can ask, well, why did they use that distance? Well, it's a distance which polarizes, and if you're trying to prove clustering, it's a good distance to use. But there are lots of choices. Bray-Curtis, you know, is a much more standard one. Yes, Ian?
That's the problem, though, isn't there? You know, for those of us who aren't statisticians, there is all this choice. You know, we need the guidance to say you should be doing this, this, or this. And, you know...
Well, instead of doing p-values, we're going to do reproducible research. We're going to be open. You have a lab notebook which says what you did, and you say it very clearly. And that's good practice in biology. Well, you have the same thing. You have some kind of notebook. You have a record of everything that you did.
So that reminds me of another question I had. I mean, if you want this really for this to work properly, the investigators would have to report every analysis that they did, as well as the one.
Well, that's the draw effect. You're not going to change that. That's the file draw effect, and then you'd have to do a meta-analysis. Yeah. Of course, you know, people only publish what they think is significant, right? So then you have all the studies which didn't show, where you don't publish a negative result. So,
Related to this, there's some few efforts to do encourage people to publish what they call executable papers. I think I can remember a couple of publishers who did that, and also, for instance, mathematical notebooks and something like that. Do you have any experience in this?
Well, the thing is, for instance, with PLOS, with this paper which is going to appear in PLOS Computational Biology, the supplementary material is enormous because it goes through every possible thing that we did in order to create every plot and so, but the problem is the overhead is enormous. That is, we had the idea of doing this normalization more than 2 years ago, and the paper is going to appear next week. And so the overhead, I can do that, right? But maybe somebody who's younger and is in a hurry is not going to do that. And the biologists, you know, they don't even want to talk to you. Right? So I work on 2 time scales. I work on the biology time scale is in 2 weeks we need to publish. And on the methodology time scale, it's different. And so that only works because I use a system which is up and running. So we work with this shiny framework where you generate web pages every time you do an analysis. And so everything is that's your notebook. And we collaborate that way. So, but you already have to have a system to do it.
One last question, Eddie.
Yeah.
I think it's an important issue. I don't think that the lab notebook is a good metaphor
Okay.
for this, because you write your experiments, the ones that don't work, you move on, do it a different way, and that's why you publish the one that worked. And so I don't know.
But it's just keeping a record. What I mean
You're
about
right.
you write down on the lab, you know, which day you did and who did it.
Right.
Those are all very important.
You don't publish this. This is in your.
No, you don't publish it, but you are, if there's some kind of contentious issue about your work, you know, you made up the data.
Right.
You have to be able to, it's like your, you know, the IRS, that you're supposed to keep your records, right?
But aren't you presenting that you should include that with, you know, the various analyses you've done?
You have to include enough of it that somebody could reproduce your pipeline and see your choices. Of course, you're not giving everything, all your code.
Just the final choices. Just the choices that actually you generate.
No, I think it's a different thing. I think this you should include.
Yeah.
Experimental.
Yeah, no.
Right.
I'm fine.
Okay.
I'm just saying there are standards that we should try to be working to, and this is one of them. So.
Okay. Well, thank you so much. So we better stop there, and we will have a lunch break now, and please come back at 1:45 to continue with our afternoon session.